Hello, everyone, and thank you for joining us today for our return of Native Voices STEM. Today we'll be talk from Dr. Conroy-Ben on wastewater-based epidemiology in tribal communities. Before we go and kick things off, just want to start very quickly with a land acknowledgement. We respectfully acknowledge the University of Arizona is on the land and territories of Indigenous peoples. Today, Arizona is home to 22 federally recognized tribes, with Tucson being home to the O'odham and the Yaqui. Committed to diversity and inclusion, the university strives to build sustainable relationships with sovereign Native nations and Indigenous communities through education offerings, partnerships, and community service. Just a little bit about our seminar series. So the Native Voices in STEM seminar series is co-sponsored by the Indigenous Resilience Center, the University of Arizona, Sloan Indigenous Graduate Partnership, the Water Resources Research Center, the Native Fuse Alliance, and the Office of Societal Impact. Seminar series highlights Native scientists, engineers, educators, leaders, and social activists, providing students with an opportunity to network and learn about the research and careers of Native leaders in various fields. Each session includes a Q&A segment at the end where all attendees are encouraged to engage with the speaker, ask questions, and participate in the discussion. So to begin today, I'd like to introduce Dr. Conroy-Ben.
No, I think I read a paper
originally from the
on
Pine
how
Ridge
GIS can be incorporated into monitoring.
First-generation college student, she went on to receive a BS in chemistry from the University of Notre Dame and an MA in chemistry, MS in environmental engineering, and PhD. Her postdoctoral co-appointments were in biochemistry and in environmental science, also from the University of Arizona. She is currently an associate professor of environmental engineering at ASU, where she researches wastewater treatment and pollution, tribal water quality, mining remediation, wastewater-based epidemiology, and bacterial multidrug resistance. Dr. Conroy-Ben teaches courses in environmental engineering, environmental organic chemistry, and soil and groundwater remediation. And now we will turn the floor over to Dr. Conroy-Ben.
For the introduction, and I want to thank University of Arizona and the Indigenous Resilience for inviting me to speak at the Native Voices in STEM, and also thank you to the partners. So today I'm going to talk about the bulk of my research that occurred during the COVID pandemic, and that includes wastewater-based epidemiology, specifically in tribal communities. I did much of this work with a very large team at ASU. I like to acknowledge them: Doctors Carrie Hamilton, Rebecca Munich, Aaron Driver, and Ralph Halden. You heard about my educational background. I wanted to talk a little bit about who I am. My name's Otakuye Conroy-Ben. That is Lakota for woman with many relatives. I'm from the Pine Ridge Reservation in South Dakota. That's located in southwestern South Dakota. I am one of 6 children, and my parents are shown there in the picture. I'm still very much involved in my culture. I grew up powwow dancing. I'm a fancy shawl dancer. And my husband, Colin Ben, is a program manager at Northern Arizona University at staff, and we have 2 beautiful little girls. You heard about my educational background, so I'm very much trained in chemistry, biochemistry, environmental science, and environmental engineering. So I spent almost 10 years here at the University of Arizona. I love Tucson. I miss Tucson. It's always great to come back to campus. I really wanted to highlight that, yes, I was in academia quite often, but I did get some real world experience. After college, I worked at the University of Colorado in a sequencing lab. And then I switched trajectories from chemistry to engineering. I wanted my chemistry to really be applied. I wanted to design strategies to clean up the environment. While I was here, I did a summer internship at the US Geological Survey in Denver, and studying methods in analytical chemistry. After I graduated with my PhD, I moved on to the LA County sanitation districts and worked as an environmental engineer, specifically a wastewater engineer. Came back to do a postdoc. While I was a postdoc, I adjuncted at Pima Community College in mathematics. I moved on to the University of Utah and then found my current position at ASU. And a long career. But I am an environmental engineer, specifically a wastewater engineer. And this kind of highlights where I've been in my life, starting with the upper left. This is, or was, the Sweetwater Recharge Facility over on Roger Road, just west of I-10. And this is where I did my PhD work. So this was early 2000s. We were looking at reusing wastewater as a water source. And there were a lot of unknowns back then, and some knowns. But we were looking to see if, after cleaning up wastewater, were there pollutants still remaining in that wastewater that could impact the endocrine system? Endocrine system is made up of your thyroid, pancreas, developed a metabolism, in addition to sexual differentiation. So in my studies, I was looking at how these pollutants get removed and then how they get discharged back into the environment. So this was really pivotal in shaping my career as a researcher, because I saw that my work was making an impact in the environment, as well as for animals and for humans, protecting their health. And I also started to really love to do research. So then I moved on to Los Angeles County Sanitation Districts in the lower left. I worked at specifically this treatment plant, JWPCP, Joint Water Pollution Control Plant. This is one of the largest wastewater treatment facilities in the world. We measure these by how many gallons of water produced per day. And this one produces or treats 400 million gallons per day before it gets pumped into some underlying pipes and discharged into San Pedro Bay. While I was there, I really missed the, I guess, freedom of academic research. And so I went back, did a postdoc, went on to my faculty position, where I now look at individual wastewater treatment processes, like the upper right-hand side, that's a wastewater treatment plant. This work has taken me to indigenous communities. In the lower right-hand side, I had the opportunity to travel to New Zealand to work with a Maori community on looking at the impacts of wastewater pollution on Lake Rotorua. And currently, one of my tasks during this trip to U of A is I'm also delivering some biosolid samples to another lab here at U of A. So I have some partnerships with NAU and U of A, where we're looking at pollutants found in biosolids that have been dewatered. These, the ultimate fate of these biosolids are to be land applied for fertilizer. So those are really the main aspects of wastewater, right? We're treating human waste. We end up trying to reuse the product for something beneficial. We have to make sure that all of the different pollutants get removed to an acceptable level. There's another area of wastewater called wastewater-based epidemiology, where we're only looking at what's coming into the wastewater treatment plant. Well, what comes in is what is collected from human waste activities. So showering, washing our hands, laundry, sanitation, in addition to what we excrete in our waste products in urine and in solid waste. So all of that waste makes its way to a treatment plant, and we're able to measure the different components in that wastewater. And we can even measure health-based indicators. And so that's what is really what WBE is all about, or wastewater-based epidemiology. So we can measure different components, including drugs. We ingest it, we use the facilities, it goes to a sewer system. We collect that sample, analyze it, and then try to back calculate what is coming from the population from where we collected that sewer sample. We know that drugs, for example, can be detected in urine. And we have analytical methods that can actually quantify what those drugs are. We know that urine is about 1% of the volume of wastewater. During the pandemic, we also learned that pathogens are shed in fecal matter. So we can measure chemicals and we can measure pathogens. When we measure chemicals, we need some type of chemical analytical technique. Typically, a mass spec instrument is required. In this case, we're using liquid chromatography to separate all the components of the different drugs, and then we analyze its mass and quantify it. We measure drugs of abuse. Other health indicators, for example, if there is some type of biomarker protein that is excreted primarily from people who have diabetes. We can also measure peptides or amino acids from these different microbes that we're trying to track. We have the ability to quantify pathogens. So pathogens are those disease-causing microorganisms. And the analytical techniques we use are qPCR, or quantitative polymerase chain reaction, in addition to sequencing. During the pandemic, started to look at coronavirus, and I'm branching out into some other areas of WBE, including looking at targeted pathogens such as E. coli, other viruses. And then we can also look at non-specific pathogens. So just taking a sample and sequencing the whole thing to see what is there. And I'm primarily working with tribal communities. I've been doing this work since 2017. And since then, I've outreached to about 44 different tribes. And each tribe has their own requirements as far as research goes. In some cases, there are tribes that don't have research policies. But I was invited by 12 different tribes to submit an application, and those could be either their own research application, a proposal, a presentation. It really varies. And the time for a tribe to make its decision also varies. I can get a response sometimes within a week. And sometimes it goes on years because it's really important to develop those tribal relationships. We have to build that trust and make sure that we're giving back to the tribal community. We're not just taking their data. So it does take time to really work with the tribe and so to date, we've received 5 full approvals to do different type of work to monitor drugs as well as pathogens. We did receive a lot of rejections when it comes to this work. There are some complicated water rights. Whenever we're dealing with wastewater, wastewater has to follow the Clean Water Act, which protects surface water. And each wastewater facility has to have a permit to discharge wastewater into the environment. Well, if you're discharging that wastewater into a large common body of water, such as the Colorado River, for example, you start to pull in other federal agencies, other jurisdictions that do require different levels of approval. And that's what really, complicates the picture. Some tribes don't have their research IRB code established. I had some tribes that didn't really think that this wastewater monitoring was going to enhance their monitoring of coronavirus. In some cases, we had projects that made it to a committee level and was approved. But when it was forward to their attorneys, it really didn't make it up to the executive level. So in some of our early work, we really wanted to see which tribes were connected to sewer lines. So this paper was published in 2022, where we looked at the 10 most populated tribal communities. And those are listed here, Navajo all the way down to some tribes here in Arizona. The states that we cover, the reservation size, population, and then the population captured by wastewater. In addition to the percent that's connected to wastewater and the number of facilities that there are. So we were able to go to some federal databases and see where these tribes are discharging wastewater through federal permits. And once we know the locations, we're able to map where these locations are. So you see a map on the right-hand side of wastewater facilities. WWTP is a wastewater treatment plant located in the United States and in Canada. So to work with these 5 specific tribal communities, we had to go through either IRBs or research review boards. Some tribes had resolutions. There's always an MOU or MOA involved. In some cases, an application and a full proposal. ASU has a tribal research review office where any faculty member who proposes to do work with tribes, that proposal has to get reviewed to protect the tribe as well as to protect ASU. So once we have all the approvals in place, all of the contracts in place, we then need to coordinate with the tribal utility. So what's unique about this work is, the health departments are interested in the data, but we also have to work with the environmental and water divisions. And those 2 entities don't always communicate with one another. We have to identify where to sample. I'm usually in the field doing that, pulling up manholes, looking how it is, looking at flow rates and so forth. And we analyze the sample, take that to ASU, and then we return all data back to the tribal community. So in the first application of WBE that my team looked at, we were interested in drugs of abuse. So this was work done with Dr. Rolf Halden. He's a very nice LC, MS. He's a very nice analytical lab that has the ability to measure different drugs in wastewater. So substance abuse is a problem in tribal communities. And pre-pandemic, we saw a serious crisis when it came to opioids. We can look at surveys to see what is going on in different communities, but surveys are not always reliable. You're only sampling a small subset of the population, and even within that subset, we're really not capturing what's going on in tribal communities. But what we do know in this graph here, this shows that the deaths as a function of time when it comes to opioids. Overdose deaths have been increasing since the year 2000. And we're dealing with fentanyl that's being laced into drugs. And so there is a need to see what is truly going on in our communities. So we're able to take the wastewater sample, process it for analysis on drugs. We collect a wastewater sample and we collect it over 24 hours using an auto sampler. So we lower this into a sewer line. We don't go to a wastewater treatment plant because those samples have already been treated to some level. We need to know what is a raw sample and what does it look like. We put that sample through some type of resin that pulls out all of the organic molecules, and then that gets injected into an instrument that will tell you what that chemical is and what is the quantity. We know the wastewater flow. We can calculate the mass of that drug that comes into a wastewater treatment plant per day. And there are known, I guess, excretion values and I guess metabolite conversions of that chemical, or we can back calculate to see the per capita drug consumption. And so I went to a tribe and I proposed to look at drugs in their community using wastewater. And they were really excited about this because it was a non-invasive sampling of their community. And we were able to get information on many different types of drugs. So at the time, this was the list of analytes that could be quantified in my co-investigator's lab. So you have the ability to look at over-the-counter drugs, recreational drugs, prescription, opioids and withdrawal medication, as well as illicit substances. And what we ended up doing was on this reservation going to several different wastewater facilities, collecting either grab or a composited 24-hour sample, taking it to the lab and then quantifying it. So what the plot on the right-hand side shows you is the mass of drug that is entering that site per day. So entering the wastewater treatment facility. And the y-axis, you have very different orders of magnitude. Site 3, for example, had very small drug loads into the wastewater facility compared to site 1. And that was also reflected in just the population and the size of the community we were from. So this tells us kind of the magnitude of what's going on in each community. Also told us what drugs were present in the community and what we actually measure in this type of analysis is the metabolite. So your body is going to convert that drug. And so what's measured is the metabolite in addition to the parent drug, and that will distinguish what has passed through a body versus what has been just dumped down a drain. And this plot really shows you the details of the drugs and the cumulative load over the whole reservation from the sites we sampled. The x-axis is a log scale mass per day of drugs. So the larger the value, that means more drug is coming into the sewer system. So we see high levels of morphine, HC, marijuana, caffeine, alcohol, and so forth. So you can see those details here. So how we got to this value is if we know that concentration from that mass spec analysis and we know the flow rate, we can just multiply the two to get that mass per day load. So these results were returned to the tribe, and the IRB was very excited to see these results, and they wanted us to do some more work. And this was before the pandemic. And when we compared our results to what is known nationally about drug use in Native communities based on surveys, what we're able to show is that we can get down to, through chemical analysis, drug-specific usage, just using mass spectrometry. And where we know the population where we're sampling from, so we could tally up the number of American Indians that we actually surveyed. And what we surveyed, the population we surveyed from exceeded the survey numbers from an actual questionnaire that was sent out by different organizations. BRFS is a national alcohol and tobacco survey, TEDS, and NVIS National Substance Drug Use. So we're able to get that granularity. We're able to survey more American Indians. We're able to see what is actually being used. For this tribe specifically, the community requested us to monitor some chemicals that were not originally on the list. So these included dextromethorphan, which is a cough syrup ingredient, and tramadol, which is a prescription pain drug. And that was because they suspected that these two were being highly abused in the different communities. And so the lab developed a method for those, and we're able to verify that, yes, high loads are coming through the sewer system. This was pre-pandemic, and this is actually before fentanyl. If you take a look here, there were low loads of fentanyl at this time, 2017-ish. And so at that time, there were elevated levels of methamphetamine and cocaine, in addition to opioids. It will be interesting to go back and sample now that we know that there's this high use of fentanyl nationwide. We also saw elevated just pain medicine in communities that had Indian health service. And we also saw trends that mirrored what's going on nationally when it comes to nicotine and caffeine. We're always going to find those in the sewer system, and those can actually be used as population indicators if we don't have that information. And then we also found ethanol consumption in this community, in addition to other drugs. Switching gears, so we conducted a drug study, we wrote up the paper. This tribe allowed us to publish those results as long as we de-identified the tribe. And as long as they had approved the manuscript. So then, as that was going on, we have the pandemic, the COVID-19 pandemic. This is a snapshot of what had been going on as far as case counts, as well as mortality rates from 2020 to 2023. American Indians on the left-hand plot, are highlighted in this yellow line, and all other races are in the other plots. But you can see that for both case counts as well as deaths, American Indians, the American Indian population was either leading through the different waves or was near the top, particularly for deaths. In about late 2020, yes, we started to see encouragement of vaccinations. And during the period of 2021 to 2023, we saw that the American Indian population was leading the other demographics when receiving at least one dose. We saw that was important in many communities where they were seeing loss of elders, for example, and that could trickle down into loss of culture. So there was a real push to try to get our tribal members vaccinated. So as that was going on, there were some funding opportunities to look at wastewater-based epidemiology to track COVID counts. Because we were all told to stay home, not all of us had tests. Even if we had tests, there's really no way we could report everything. So wastewater was a good way to take a sample and see if we could count how many viral particles were present in a wastewater system. So we received some smaller seed funding from National Science Foundation to see if this can be implemented in tribal communities. Wanted to develop a database of tribes that had connected sewer lines. And we wanted to do some sampling and to develop those methods in the lab, work out all the kinks to try to take this to a full-scale survey. What we found in this work was that there are just over 100 tribes with these permits to discharge wastewater. There are 326 tribes with land. So meaning people may live there, they may, some of these land parcels may not be habitable, maybe using maybe other purposes. But in any case, there were over 200 tribes with land that didn't need to report sewage and wastewater use. So what does that mean? We really wanted to see if these tribes have a population living on land, what are they doing with their wastewater? And can we somehow get those samples or at least see what's going on with wastewater treatment? Covered that many of these other facilities are operated by Indian Health Service or Bureau of Indian Affairs, Bureau of Indian Education, in which case they wouldn't be necessarily found in a public database just because of the size and perhaps the design of the wastewater treatment plant. There are also some tribes that don't discharge, but maybe use their wastewater for some other purpose. So either reclamation, either they're pumping it in the ground, or maybe the facility is a terminal lagoon, so it's non-discharging. So these tribes would have facilities not present in the database. We also wanted to see, obviously, can we measure coronavirus in a wastewater sample coming from a tribal community, and how do we go about doing that? So we ended up developing a plan. We worked with a tribe that quickly approved this research because they needed to get a handle on what was going on with the pandemic. And so we were able to take that information, take those practices, and develop a large-scale surveillance of tribal communities. So we were funded by National Institutes of Health to recruit 5 to 10 more tribes in this work to monitor routinely coronavirus and wastewater. So we know that based on the previous work, 100 tribes have centralized sewer treatment in the US that discharges to surface water. And we also found out that we can collect from sewer lines, we can collect from the headworks, which is where all of the sewer enters before the wastewater treatment plant, as well as from lift stations. Our work, we ended up working with 6 tribes and collected from 12 locations. 5 of these tribes had full wastewater treatment plants. 5 were more remote, and we were collecting from just upstream of lagoon systems. Then we also had 2 locations where the tribal wastewater was intermingled with non-tribal. So we had to kind of map the sewer system and see where we can actually sample from. And so approvals, years later, we're able to get sample and start to monitor tribal communities. also point out that there was a push to see what these variants were circulating in the environment. However, when it comes to tribal research, several tribes do not allow genomic sequencing of any, and in some cases, environmental sequencing. So as we were working through the approval process, we really tried to include genomic sequencing, and ultimately 3 of our tribes allowed it. So we can see. So the advantages I already highlighted, we're collecting from a large population, and that was important at the time to look at population levels of coronavirus. This was non-invasive sampling, right? We weren't going and asking for biospecimens. A wastewater sample in some cases was considered an environmental sample. And this data can complement existing data perhaps put out by CDC or Indian Health Service. Some disadvantages as we were working through the pandemic is that we didn't know the population contributing to the sewer line, as well as the sewer flow rates. In some cases, we had low sewer flow rates and we couldn't pull up sample during all of the 24 hours we were sampling. We're dealing with remote locations that were sometimes difficult to get to. We had to coordinate and maybe work only within certain hours of the day. Conditions such as weather. And then we are dealing with human waste, which contains pathogens, so we have to take appropriate precautions when sampling that wastewater. There are some pictures of some of our field work when it comes to wastewater sampling. What is recommended by CDC is that we get an auto sampler and install it from wherever we're collecting that can be programmed to pull up for a full 24 hours. So we ended up collecting in some locations every hour, bringing those back, analyzing, and then making a composite. Because what CDC does is tell people to make a 24-hour composite sample. So this is a picture of an auto sampler. The bottom center is an installation into a manhole. So we go and pull up the manhole and set the robot in to feed tubing, pulling off the sample, and then bringing it back to our lab to analyze for coronavirus. As part of this large NIH work, we were also doing a few other things, right? Engaging with the tribes, making sure that they get their data in a timely manner. Always optimizing our analytical techniques. In addition to figuring out where within the sewer system is a good place to sample. And looking at risk associated with working with wastewater. So during the NIH portion of this grant, which started in August, September of 2020, we already had a couple tribes on board, and it took us another 1.5 years to recruit the. So for example, in 2022 alone, we processed over 1,300 wastewater samples from 4 different tribal communities. This flow diagram on the bottom really just shows you how we process the sample. So we take that raw wastewater, it gets filtered to remove all of the particulates and junk. And then that filtrate, which would contain a virus particle, gets concentrated. And we'll extract the RNA from that and then put it through, mix it with some chemicals and put it through a PCR instrument. And what was been good about this is that once we got in a sample, it's processed, analyzed, we can get results back to the tribe within 3 days. As soon as we got the sample, I'll point that out. And over the 12 different sites that we collected from, comparing to the different waves of the pandemic, we saw comparable trends. So if there was a spike, Omicron came on, we saw a spike in wastewater. And we found that for some communities where they weren't actively collecting clinical data, we were able to fill in the gaps with wastewater data. We're able to get those samples routinely for the most part, but we weren't, in some cases, due to weather conditions, staff unavailability, that infrequent sampling really limited the interpretation of this data. This plot here shows you what data comes out of a wastewater sample. So on your y-axis, this is a coronavirus copy number. So we take that qPCR that tells us how many genes per volume we have. And if there's one gene per viral particle, we say that's the number of viral particles present. This is a log scale. So we're going upwards in some cases of 1 million coronavirus particles per liter of wastewater, which compared to other sites nationwide is very, very high. And those really trended with the spikes during the pandemic. For this tribe in particular, this shows the infrequent sampling. So this was year 2022, 2023. And we're only able to collect on a monthly basis due to travel restrictions and staff availability with their tribe. So we're able to show that, yes, we have coronavirus present, but ideally we want to collect more frequently and more regularly. I had one other project associated with COVID. This is through NARCH, Native American Research Center on Health. And this is led by the Intertribal Council of Arizona and the University of Arizona. So I am a sub-awardee where I proposed to sequence those wastewater samples that were positive, that were permitted by the different tribes, and to look at developing a tribal wastewater framework, to look at perspectives. I'm going to survey tribal health departments and water and sewer programs nationwide. And then I'm using this as an opportunity to explore other wastewater applications in tribal communities, including infectious disease. So we can take a wastewater sample, use next generation sequencing, do some whole genome sequencing, deep sequencing, and really see what are the strains present in a wastewater sample. I compared what was present in that wastewater to CDC's microbial threat list, and we're able to detect a lot of the pathogenic bacteria, viruses. We're able to detect that in wastewater. And also on the radar of EPA, CDC, NIH is antimicrobial resistance. So along with that deep sequencing, looking at the strains of bacteria, we can look at the genes involved in antimicrobial resistance. So as a wastewater engineer, I'm interested in what goes on across the different wastewater processes. We could see population changes, obviously. We're able to see what the microbes are resistant to. For example, we're detecting a lot of multidrug resistance. You might hear that in the news, tetracycline resistance and macrolide resistance. And I'm going to continue that work through an EPA grant that was awarded. We're working out the contract, and I'm looking to identify tribal communities who are interested in participating. So to wrap up, we face a number of challenges in wastewater-based epidemiology and its implementation in tribal nations. We have limited staff on the tribal side, and so a lot of the work, the sampling, going out and working the manholes and wastewater treatment plants, that fell on the ASU team. Even through our travel restrictions, we had to assess that sewer infrastructure. Not all tribes have the right sewer infrastructure to do this work. If what the sample's coming from a faraway place, you have to factor in driving, FedEx, and so forth. So during that 3 to 5 day span, in some cases, the sample degraded. There were many sampling options that tribes were interested in, but we just didn't have the time to navigate the IRB approvals, particularly through Bureau of Indian Affairs. If we're trying to sample from an under 18 population, that was going to be very difficult to get that approval. Tribes are interested in casinos, and there are some subdivisions that have their own little sewer systems that we can potentially collect from. And so what did we accomplish? We have a list of tribes that we can collect from. And so if there is another pandemic, we know who to go to and where these locations are. We sampled 5 different tribal nations, about 2,000 samples over the pandemic using PCR. We have some data showing the different variants of coronavirus. And another interesting application is my co-PI started to develop a protein mass spec method for tribes that didn't allow genomic sequencing. So this is looking at a chemical signature rather than a genetic signature. My other co-PI looked at an online risk tool for wastewater exposure. So for wastewater operators who may be concerned about coming in contact with wastewater, she developed an algorithm to calculate that risk. And we're working on an analysis of tribal sewer networks. So where in a sewer shed with all the pipes is the best place to collect from. And this kind of summarizes everything we've done. So we're looking at making sure that tribes get this data back. We're working with the community, partnering with tribal colleges, measuring different health indicators, modeling wastewater treatment, looking at the risk when it comes to sampling and working with wastewater, and where we can sample from. And I'd like to thank all of my collaborators, team, tribal partners, and funders. And if you're interested in this work, please reach out to me. My email is listed at the bottom. Okay, thank you.
So thank you so much, Dr. Conroy-Ben, for taking the time to be here with us today. I think on behalf of the Indigenous Resilience Center, we're all really grateful that you would come to be here with us, share your wisdom, and just share some of the really impactful work you're doing in communities. Really insightful, I think, for a lot of our graduate students and the postdocs to see kind of like a testament to relationship building and trust building in communities with these approvals, and as we try to navigate this process ourselves. Next, what we're going to do is open up the floor for questions, which is our Q&A session. We really encourage people online and in person both to bring forward any questions you might have about Dr. Conroy-Ben's journey, her work, and just anything that you really want more insight into. And we'll now turn this over to Ruth, who will be controlling this via Zoom.
Mark, I see your hand up if you want to go ahead and ask your question.
Yeah, I appreciate you, Dr. Conroy-Ben, being here today. I wanted to know, have you worked with the San Carlos Apache tribe on some of your work already? And if so, are you willing to come out with your new grant to work with particularly our environmental department? Because now I'm the environmental coordinator. of the tribe. So I was wanting to know what's your take on that from your previous research and potential upcoming research.
So are you from the San Carlos Apache tribe?
No, I'm working there now as environmental core. I was working for the college and I've taken a new position with the tribe. So I was the environmental biology teacher, and now I'm the environmental coordinator with the tribe. So I'm working now with dealing with air quality and wastewater, water quality stuff there for the tribe now.
Um, so a few things. I did write a large NIH grant, which was to be reviewed in that was pulled. But then it was put back in. So I don't know about the future of funding for this type of work, unfortunately. If it does move forward, I definitely will reach out to tribes, particularly local, because it's easy access to wastewater. So definitely I will reach out to San Carlos
Okay.
if funding looks promising.
Okay. Where I was more particularly interested in your intertribal council grant that you were working on. Are you working with San Carlos on that one too?
No, so that grant ended last May.
Oh, okay. Okay.
And so right now I'm just looking for new funding.
Okay. Thank you. Thank you. I'll be in touch.
Question in the chat, Nancy, if you want to ask it, you can, or I can.
Thank you. Yes, hi, Dr. Conroy-Ben. It's Nancy Peterson, and I just really appreciated your presentation. And I'm thinking about what you were saying about for future pandemics, and I'm wondering how long it takes to develop the analytical techniques to identify the pathogens in wastewater, and could you use that for all pathogens, or some pathogens just aren't going to be transmitted into the sewers?
Right, so the pathogen has to be excreted in waste. And I believe the virus that causes the bird flu is excreted in waste and is being detected in wastewater. So virtually any new pathogen that is being flagged as a potentially infectious has primers designed. And once we know that sequenced, for qPCR, that method can be developed within a week.
Thank you so much.
Thank you so much for your doc. I had a question about the protein mass spec method. So has it been very successful for creating a pathway to work with tribes that don't allow any sort of genomic or genetic testing?
We have not analyzed any tribal sample that way. And we have not proposed that as an alternative yet.
Okay.
So right now they're just working on the method development.
Working on the methods.
But yes, it came about because of those reasons.
Cool. Thank you.
I have a question. In this work, do you think was one of the more unexpected challenges?
Let's see, I would say data that was not routine that we did get regularly is how do we treat that data? Because we're just reporting results back to the tribe. As researchers, it's not up to us to make recommendations, but more to kind of tell them that, okay, coronavirus looks up at this. And so I would say unexpected is we didn't really fully think about how to provide guidance on the results. Because it's, I mean, it's a whole new field, especially for tribes.
To kind of go off of what you were just talking about, have you since developed some form of methodology for explaining the results?
So yeah, so in explaining the results, we were able to say these levels are high versus low. And it is a little bit more retroactive, so their health departments were able to say, okay, yes, our clinical results are also high or low. And so we kind of got a feel as to what levels were kind of like warning indicators.
And they can develop responses based off of that information, right?
I don't know that they did. That was the goal, but weekly sampling would get us there instead of.
I guess it would be how, or how did you first become interested in the work, and then that gap between your interest and deciding to kind of pursue it?
But I become interested in wastewater.
Yeah, the field, like specifically, I guess.
So I became interested in wastewater here because the research is very, very interesting. So we're looking at endocrine disrupting chemicals in wastewater. At the time that was causing, a lot of research was coming out saying that these chemicals were causing fish to become female and populations were changing. There's a lot of interesting information in wastewater. And wastewater is an environmental problem, right? The discharge, how do we reuse? It's very energy intensive. So it's, as an environmental engineer, it's a really dynamic field to be in. Chemistry, biology, and so forth. Comes to WBE, it was just another interesting aspect of wastewater to look at.
To the work around wastewater and reclaimed water and groundwater recharge that was already being done into some, or that just happened to align with.
Yeah, that happened to align. Yeah, I actually didn't know that research was going on at U of A when I accepted.
So again, I want to thank Dr. Conroy-Ben and everybody for joining us here today. Also a big thank you to our co-sponsor. So of course, the Indigenous Resilience Center, the UA Sloan Indigenous Graduate Partnership, the Water Resources Research Center, and the Office of Societal Impact for making this seminar possible. So we'll invite you to join us for our next Spring 2025 Native Voices in STEM seminar. That would be with Dr. Marco Hatch on March 5th. Before we officially close, we want to encourage students to stay online for a more informal Q&A session. It's a great opportunity to ask any additional questions, seek mentorship, or engage in discussion with Dr. Conroy-Ben in a smaller student-focused setting. But again, thank you all, and we look forward to seeing you at our next session.